Hello, my name is Ryan Beshai. I'm a PhD candidate at the University of California, Irvine, and today I'm thrilled to be talking to you about the population impacts of two range-expanding predators along the Northeast Pacific coast. So, as I'm sure everyone is aware, anthropogenic warming is increasingly affecting species and ecosystems worldwide. And in response to warming, many species from many different taxonomic groups have begun redistributing from warmer into cooler regions. Typically, this occurs towards higher elevations or towards higher latitudes. And one of the interesting parts about this is that even though this may be necessary for global species biodiversity preservation, these range-shifting species also have the potential to strongly negatively impact their recipient communities. So an example of one of these range shifts is a species that I'll be talking a lot about today, the dark unicorn snail 𝑀exicanthinoLugubris. This species has historically ranged along Baja California in Mexico, but has recently expanded here into Southern California. And what's interesting about this process of moving from a historic to an entirely novel community is that it may remind you a lot of an invasion by an introduced species. Now, this is really interesting because we don't know a lot about predicting the impacts of these range-expanding species, but invasion biology has developed several frameworks that are attempting to try to do exactly this. And so the point of this project, the goal, is really to test whether or not we can apply invasion frameworks to range-shifting species. And in particular, we were interested in testing whether or not range shift impacts can be predicted by either the abundance of the shifter or by what the shifter does in its historic range. So to get at these two questions, we utilize the system of range-expanding whelks. So both of these species, Acanthonucella spirata, are undergoing poleward expansions. I've already introduced Mexicanthina. Acanthonucella in the dark green is historic along California and is recently expanding into northern California. Both of these whelks are known to exert fairly strong community control over community composition, particularly through their predation on foundation species such as barnacles and mussels. So, using these two species, we decided to conduct this manipulative caging experiment to really suss out the relationship between expander abundance impact, as well as to compare impacts between historic and expanded regions. So, for each species, we identified two historic and two expanded sites. We then applied the following treatments at each site. We identified 7 plots, 2 of which were controls, one without a cage, one with a partial cage, essentially to control for the effect of a roof in the intertidal. We then identified 5 plots that were corresponding to density manipulation treatments. So these ranged from what were effectively 0 whelks per meter squared to 120 whelks per meter squared, which are densities that were relatively ambient at each of the sites. We then monitored these cages every 2 weeks for 8 weeks, during which we conducted community diversity surveys looking at prey and diversity, as well as maintaining whelk density treatments in case we had any escapees. We then looked at 2 particular responses. We were interested in first the loss of living barnacle cover, as well as predation or loss of living mussels. And these ones were easy to tell because whelks leave a very distinctive drill hole on mussels that they've predated upon. We then took a 2-step analytic approach to address each of these questions. First, we determined the abundance impact relationship using linear and quadratic regressions to kind of determine whether we were following a linear or nonlinear abundance impact curve. We then contrasted the impacts between historic and expanded ranges using linear mixed effects models. So what would we predict to see? Well, if we're going to draw on predictions from invasion biology, we can plot whelk density on the x and then our response on the y, and we think that this would follow that similar pattern to those of invasive predators where we see a nonlinear response or increasing impact that then saturates at a certain density. Then, if we think that the impacts in the historic region are going to predict those in the expanded region, we expect to see very similar responses at each of our sites. So, how does this actually translate? What did we find? Well, so I'm going to show you a series of plots. Each plot will have 4 panels. The panels on top represent sites in the expanded range. The panels on the bottom are sites in the historic range. Then, for each of these panels, each point represents a cage. So here we're looking at Acanthonucella, impact on barnacles. And in this case, we can see that it has a very clear linear increase in impact on barnacles, regardless of where this is in the range. In contrast, now we're looking at 𝑚exicanthinA on the barnacles, and here this was really interesting because we didn't see a similar consistent linear increase regardless of range position. We found it in the historic range in Mexico, but we couldn't find any impact increasing with abundance in Southern California in its expanded range. And this was very odd because we couldn't figure out why we wouldn't see a greater predation impact with more predators. So we started thinking about it a little bit more. And if you take a look in the bottom left-hand portion of this quadrat, you can see barnacles that are basically just sitting on the bare rock. Then you can see some in the top right-hand corner where they are distributed on top of mussels, which are second preys. And we thought there might be an effect of kind of refuge provisioning from the mussels on the barnacles. So in other words, we think that the ones on the bare rock are going to be consumed faster. So we had an undergrad who went out and did essentially this, reclassified and then reran each of our models, and essentially found exactly what we thought was happening, where we're not seeing a lot of change on the mussels, on the barnacles that are on the mussels. They tend to survive very well, but barnacles that are just sitting on the bare rock, we're starting to see that relationship emerge where higher density of the predator leads to higher impact. Then, with our final predator-prey contrast, we're looking at 𝑚exicanthinA on mussels. In this case, we can see again that the impact is linear and it is increasing at all the sites that we were able to assess. We weren't able to look at this relationship in Punta Moro because mussels didn't occur very frequently in those plots. So again, this consistent linear abundance impact curve. Then, when we take a look at the relationship across space, so between historic and expanded regions, what did we find? Now we're looking at 3 plots. These plots each kind of summarize what we've been looking at before. So here, each point represents a slope from those 4-panel plots. The bars represent 95% confidence intervals, and the asterisks or plus signs represent significance of that relationship at the 0.05 or 0.1 level. So here what we're seeing is that for Acanthocella, regardless of where it is in its range, whether that's its expanded range in Mendocino North or South, or its historic range in Dana or Scripps, we're seeing very consistent per capita effects. These are the effects of any individual whelk on loss of barnacle cover. We're again seeing something very similar for 𝑚exicanthinA, even though the relationships were not significant. We could still determine that slope, what it would have been if it was significant, and we can see that that slope is consistent across ranges. The big difference that probably popped out on the page on that previous slide is that for 𝑚exicanthinA on mussels, here at panel C, we can see much, much greater per capita effects of 𝑚exicanthinA on mussels in their expanded range than in their native range. And this suggests that generally these per capita effects are conserved, but that they might be slightly larger in the expanded range. So what does this kind of all mean? What does it lead to? Well, we can see clearly that these overall impacts may be predicted by shifter abundance, but that can be difficult to resolve. And that is particularly true for generalist predators in the sense that if you're eating one species, it may be difficult to determine that abundance impact relationship on a secondary prey species. Second, this general per capita conservation with the potential for greater per capita effects in the expanded range, I think is really important from a management perspective in terms of anticipating species that are likely to have the largest effects. So for example, if you see a species that you may want to manage, a range shifter that you may want to manage, you may want to target ones that already have really large impacts in their historic communities, thinking that they may be similar or slightly larger in their expanded communities. And then finally, I think it's really interesting thinking about this linear impact abundance relationship. So this also has really important management implications because it suggests that if you aim to reduce densities of the expander from the highest point to somewhere lower, you're going to have a large effect. Whereas if we were looking at a nonlinear relationship, you'd want to do this much earlier in the expansion or invasion process, whereas this is much more feasible to implement. So if you only take one thing away from this presentation, I'd like you to remember this. It's that it does appear as though we can use these invasion frameworks to predict range shift impacts, specifically both the abundance of the shifter or its impacts in its historic range can be used to anticipate impacts in its expanding range. Finally, I'd like to thank my collaborators in Northern California and Baja. This project would be impossible for me to have done on my own. I'd like to thank all my lab members for their support and my advisor as well for her contributions. I'd like to thank the Department of Education for funding as well as the National Science Foundation. So thank you for watching, and please feel free to reach out to me with any questions. Thank you. Bye.